So students, welcome back. So we did about yesterday from the first day of our session, understood quite basics about biochemistry, about DNA, RNA, nucleic acids, various amino acids we have discussed in details. We have discussed about PCR, we have discussed about DNA isolation and their virtual labs also. So today we will do a little bit in first hour subjective. In the next hour we do the virtual lab of southern, northern, western digital electrophoresis, blottings. The first thing is something related to nucleotides, your pyrimidines, biosynthesis, how it is done. If I'm having some problem today with the streaming, at the moment it's going excellent, but sometime the streaming could go down. So please text me. I have turned on my text also in order to see it. So yeah. Text me on Viber if something is wrong. So biological functions of nucleotides, they're the building block of nucleic acids. They're involved in the energy storage, muscle contraction, active transport, maintenance of fine ingredients. They're activated intermediates in biosynthesis, and they're components of coenzymes such as NAD+, FAD, FMN, coenzyme A. They're also metabolic regulators of secondary messengers like CAMP, CGMP, phosphate donors in signal transduction, and the regulation of some enzymes via adenylation of and uridylation. This is a typical structure of nucleotide, including phosphate, pentose sugar, and purines or pyrimidine base pairs. And these are various nucleus base structures. Purines are including your adenine, guanine, xanthine, hypoxanthine, and the pyrimidines includes your cytosine, thymine, and uracil. So two major routes for nucleotide biosynthesis. One is salvage pathway, another is de novo pathway. The salvage pathway includes your activated ribose like PRPP plus base give rise to nucleotide, and then DNTPs could be removed. In the de novo pathway, you have your activated ribose plus amino acids plus ATP plus carbon dioxide, then it's giving out to nucleotides like DNTPs. For example, in the synthesis of pyrimidines, so this is the first section. So you have a bicarbonate in the presence of glycine and ATP. It produces glutamate, ADP, and phosphate. Then your carbomoyl phosphate is made. Then aspartate is able to remove the phosphate group. Then become you have a carbomoyl aspartate, which give rise to dihydroorotate, and further orotate, and then orthidine monophosphate. So this further in this OMP, like orthidine monophosphate give rise to uh, unimonophosphate, and then CMP, cyclic monophosphate. Then the CMP, UMP together they make cytidine and then uridine. Then from uridine, uracil is produced. Then high dihydrouracil. Then uridopropionate. Then beta-alanine. Then malonate, semialdehyde. Then acetyl coenzyme A. Then there are some defects in de novo pyrimidine. That is biosynthesis that leads to clinical diseases. Orotic aciduria. The symptoms are like anemia, growth retardation, aortic acid excretion in the body, and the reason behind it is defect in the phosphoribosyl transferase or orthidine decarboxylase. So this is the reaction showing about orotate with the PRPP 5 phosphoribosole 1 pyrophosphate, giving rise to autodiol and uridylate. The treatment is patients are fed with uridine, and UTP inhibits carbamyl phosphate synthase too, preventing the biosynthesis and accumulation of orotic acid. Then UTP inhibits carbamyl phosphate synthase too, preventing the biosynthesis and accumulation of orotic acids. So glutamine plus hydrogen carbonate plus ATP give rise to carbamyl phosphate, carbamyl aspartate, OMP, UMP, UDP, UTP, and CTP. So carbamyl phosphate synthetase. So in summary, what we have discussed is the pyrimidines are synthesized by de novo salvage pathway. The pyrimidine ring is synthesized from pre-assembled ingredients like carbamyl phosphate and aspartate, then attached to the ribose, and then pyrimidine biosynthesis is tightly regulated via feedback inhibition, and the mammalian enzymes are multifunctionals and form multi-enzyme complexes to increase efficiency. Further, drug inhibitors of pyrimidine biosynthesis are under development as potential antimicrobial and anti-cancer agents. With some references. Nucleic acid biosynthesis. Now let's discuss about enzymes. So to familiarize, now I will make you to understand how enzymes are act as a biocatalyst and study their types and their structures, and the mechanism of enzyme action, what is the specificity of enzyme and its function. So enzymes are proteins that contain that increase the rate of reaction while lowering their energy of activation. So they catalyze nearly all chemical reactions taking place in the cells of the body, not altered or consumed during their actions, and they are reusables. So first thing first, what is active site in the terms of enzyme? So active site is the area of enzyme where the substrate or other or substrates attaches to it is called as active sites. So enzymes are usually very large proteins, and the active site is just very small regions of that enzyme molecule. So these are the active sites. So enzyme molecules contain special pockets or cleft called active sites. Here we can see the green substrates when attached to the enzyme. On the active site, after attaching, it will make an enzyme substrate complex. Then after making enzyme substrate complex, this enzyme product complex is made, and then with the help of that, your products are moved afterwards. An enzyme could be used million times within a minute and produce lots of products on a continuous basis. And there is a lock and key model. In the lock and key model, it says that the activity site has rigid shape. Yeah, only substrate with a matching shape can fit in it, and the substrate is a key that fits to the lock of the active site. And this explains enzyme specificity, and this explains the loss of activity when enzymes denatures. So lock and key analogy: you have a key substrate and lock enzyme, and correct fit will react to it, and if it is incorrect, then no reaction. So apoenzyme and holoenzyme, the enzyme without its non-protein moiety is termed as apoenzyme and it's inactive. And holoenzyme is an active enzyme with its non-protein component. So apoenzyme will attach to the cofactor, and that cofactor will be a coenzyme, as apoenzyme will be inactive. But when it is there, apoenzyme and cofactor, they come together, it's become a holoenzyme, which is an active state, and a substrate could attach to it. So there are two types of cofactors. which is a non-protein chemical compound which attaches to enzyme and is required for the catalysis. That is your coenzyme and prosthetic groups. So the non-protein component loosely bound to apoenzyme by non-covalent bond. And examples are vitamins or compound derived from vitamins. Prosthetic groups, the non-protein components that are tightly bound to the apoenzyme by covalent bonds, is called prosthetic groups. So enzyme specificity. So enzyme have varying degrees of specificity for substrates. They could recognize and catalyze single substrate or group of similar substrates or particular type of bond. So activation energy. So basically, a chemical reaction requires some amount of energy to get them started. So it is first pushed to start the reactions, and this energy is called as activation energy. So this is without enzyme. You push some energy, your lactose, you want to break these lactose bonds, and in order to broke those bonds, glucose and galactose would be separated. So activation energy without enzyme and net energy released from splitting of your lactose. So structure of enzymes, it is of two types: complex or hollow enzymes and simple only proteins. An apoenzyme could be of two types. Your hollow enzyme will be divided into two parts: apoenzyme and cofactor. Cofactor would be of two types: prosthetic groups and coenzyme. Prosthetic groups includes usually small inorganic molecules or atoms, usually tightly bound to apoenzyme, where the coenzyme is the large organic molecules loosely bound to your apoenzyme. Mechanism of action of enzymes. So enzyme increases reaction rates by decreasing the activation energy. Enzyme substrate interactions, formation of enzyme substrate complex by lock and key model and induced fit model. So first thing that happen in enzyme reaction is enzyme plus substrate, they make enzyme substrate complex. So within the active site, ES give rise to E plus P, and then afterwards product is formed. So this is the short reaction. E plus S give rise to ES and then E plus P. An enzyme substrate complex in the step 1, it has enzyme and substrate combined to form a complex. E plus S give rise to ES. In the step 2, enzyme product complex is formed. Yeah. And between is the transition state. Then enzyme and product separates. Enzyme is ready for another substrate. So in this study, we have studied the complete details of enzymes, how enzyme act as a biocatalyst, and explain the concept of enzyme action. Thank you. That was short. A little bit introduction about enzymes. Ooh, lots of vaccination centers. Okay, now let's continue with next part. Further, how enzyme works. So we will discuss the nomenclature and classification of enzyme substrate complex, cofactors and coenzyme, and proenzyme. So basically, your enzymes are divided into 6 parts, that is oxidative, transferase, hydrolase, lyase, isomerase, and ligase. And oxidative catalyst redox reactions includes your lactate and pyruvate, including lactate D. In the presence of lactate dehydrogenase, they are doing the reductase and oxidation reaction. So your OH group is removed, H is removed, and mainly COO is left. In the transferase, one group is transferred to another. So in transminases, they transfer the amino group, and the kinases, they transfer a phosphate group, like this. So like in norepinephrine, methyl group donor is there. With the help of PNMT, epinephrine is made, and the methyl group has been attached. Then hydrolases, which cleave bonds by adding water, like phosphatases, peptidases, lipases. So you add 3 molecules of water, and then you add the lipase enzyme. It will remove all the oxygen bond from here, and you will have 3 molecules of fatty acids and lysases, which remove the groups to form double bonds on the reverse of double bond. So decarboxylases synthesis, you have a fumarate plus H2O give rise to fumarase and malate. Then the isomerases, which catalyze intramolecular rearrangements, like apomerases, mutases. So we have 3-phosphoglycerate. This phosphate group will move from third carbon to the second carbon, 2-phosphoglycerate, in the presence of phosphoglycerate mutase. Then in the ligases, they catalyze a reaction in which they break or make bonds between carbon, CO or sulfur, carbon, oxygen, or carbon, nitrogen. So there's a DNA strand plus 3' OH and the phosphate group 5' DNA strand, giving rise to DNA ligase. In the presence of DNA ligase, these two DNA strands are joined together. So how we give them name, the nomenclature of these enzymes. So in most cases, enzymes names end in A's. So if urea is there, its end name is A at the end. So what we do is we just replace with A's. So urease. Lactose is there, then O's, then lactase. So other enzymes are named like lactate dehydrogenase, pyruvate decarboxylase, and some names are historical, which doesn't have any relationship to substrates like catalase, pepsin, chymotrypsin, trypsin. The effect of enzymes on the activation and energy of a reaction. So there is a K, a constant K equilibrium, which will be if the reaction is small a and capital A, then small b and capital B. Then product raised to power small b, reactant raised to power small a. So enzymes speed up the reaction by lowering the activation energy, changing the reaction pathway, and this provides a low energy route for the conversion of the substrate to products. And every chemical reaction is characterized by the equilibrium constant, that is K equivalent, which is a reflection of the difference in energy between reactants and products. So you have a diagram of energy difference between reactants and products here. You have an uncatalyzed reaction and catalyzed reaction. So uncatalyzed reaction, the activation energy is quite high. That is your reactant A plus V giving you product C plus D. The activation energy seems quite high, but in the presence of catalyzed reaction, in the presence of enzyme, this activation energy becomes very lower. Beyond that, the rate of uncatalyzed reaction, on the left hand side, we can see the substrate's concentration increases as the rate of reaction increases. But in the second case, in the enzyme catalyzed reaction, two stages are happening. First, enzyme substrate complex is formed, then it is slowly converting to products. And the rate is quite limited by the enzyme availability. And we see hyperbolic graph here. Reaction reaches at maximum rate at the later, which is called as Vmax. Then comes enzyme substrate complex, which has enzyme plus substrate give rise to enzyme substrate. Then there's a transition state, enzyme product complex, then enzyme plus product. So these reversible reaction steps present the steps in any enzyme catalyzed reactions. The first step involves formation of enzyme substrate complex. ES is the transition state, EP is the enzyme product complex. Further, enzyme substrate complex details are shown here. That the part of enzyme that combine with the substrate is the active state site. Just a second, students. Give me a second. Only two students are here. Only two students have joined. I don't know where the rest of the members of the class. I think no one is interested. That's very sad to hear. Anyhow, I will give my lectures. So these active sites includes your pockets, cleft. They change or in the shape of substrate, and they hold the enzyme attracts and hold the enzyme using weak covalent interactions, and conformation of the active site determines the specificity of the enzyme. Lock and key enzyme models: enzyme plus substrate, giving you enzyme substrate complex. And in this model, your enzyme doesn't change shape, but in the induced fit model, enzyme take a conformational change. So in this transition state and product formation, how does the enzyme promote a faster chemical reaction? So as a substrate interacts with the enzyme, its shape changes, and this new shape is less energetically stable. And this transition state has features of both substrate and products, falls to yield product which dissociate from the enzyme. So like this, in this state, we have glucose and fructose. So what happened that the uh, this H2O group is removed, water is removed, and they come together to make sucrose. Same way, enzyme substrates are there. They have the functional groups, so they come together, water is removed, and they join together like this. Then possible types of transition state changes. Enzyme might bring two reactants into close proximity and maintain proper orientation. And third is enzyme might modify the pH of microenvironment and donating or accepting the hydrogen ions. So cofactors and coenzymes. So apoenzyme plus substrate give rise to no enzyme substrate complex, no reaction. Then cofactor will be attached to it as a copper ions, then enzyme reaction will takes place. So we can also see that coenzyme is required by some enzymes. So an organic molecule bind to enzyme by weak interaction. So we have a coenzyme here, an enzyme here, and then substrate S2, functional group is there, and substrate S1 is there. So what will happen? Coenzyme picks up functional group from substrate 1, and coenzyme transfer to the another substrate. And then you have a new products, P1 and P2. Whereas P1 functional group is transferred to the P2 one. So some water-soluble vitamins and their coenzymes, that is your thiamine, riboflavin, niacin, pyridoxine, folic acids. So all these having these coenzymes and their various functions. This we do not, okay. This is nicotinamide adenine dinucleotide in oxidized and reduced form. So NAD plus and NADH. So how the H group is removed, hydride ions is removed in order to make them oxidize and reduce form. This we can see from this figure. Further, NADA plus to NADH mechanism. So nictamide part of NAD plus accepts the hydride ions from the alcohol to be oxidized, and alcohol loses its protons to the solvent. So left side is the oxidized form, and the right hand side is the oxidized reduced form. In the first case, our nictamide doesn't have hydrogen ion, that's why it's called oxidation. And then when it has, then it's become a reduction. It's a reduced form. And there's the allosteric enzymes. They are the ones which change the activity of enzyme by binding at a second site. So some factors speed up enzyme reactions. Like they could be positive, which speed up, and they could be negative, which slow down the reaction. So you can see in this diagram that is in the A, we have a factor binding site and enzyme, and when substrate is attached, hence the product is produced, which will act as a negative feedback factor. It will come and attach to the factor binding site. After attaching to the binding site, your active site will be closed. So this whole mechanism is known as allosteric mechanism. So this whole mechanism that we have seen is known as allosteric mechanism. So in this reaction, we can see a third reaction of glycolysis places a second phosphate on fructose 6-phosphate. ADP is a negative factor, AMP is a positive factor of the enzyme phosphofructokinase. So we have fructose 6 kinase plus ATP and phosphofructokinase. So you will have fructose 1,6 biphosphate plus ADP. Further, in the feedback inhibition, allosteric enzymes are the basis for feedback inhibition. With feedback inhibition, product late in series of enzyme catalyzed reactions serve as inhibitor for previous allosteric enzymes earlier in the series. So you have A, B, C, D, E, F. So F could be attached to the enzymatic allosteric site of E, which will not let it active, and no substrate could attach like B to it, so no reaction will take place. So F is serve as to inhibit the activity of E1. So F is act as a negative allosteric effective. Then there are proenzymes. What are the proenzymes? They are enzymes that made in the active form. So they are inactivated in active form, but by proteolysis. So when needed at the active site in the cell, pepsinogen is synthesized and transported to the stomach, where it is converted to the pepsin. So proenzymes of the digestive tract. So it's a proelastase, trypsin, trypsinogen, chymotrypsinogen, pepsinogen, carboxypeptidase. So they will produce various enzymes. Protein modifications. In protein modifications, a chemical group is covalently added to remove from the protein. So covalently modifications either activates or turn off the enzyme. The most common form of protein modification is addition or removal of phosphate group like serine, threonine, tyrosine. So let's discuss about inhibition of enzyme activity. But I can see mainly one student is attending the class or two students, but not much. I feel quite low in order to continue class. Could you please, students, tell me your name, who is in the class in the chat box? I would really appreciate that so that I could know at least those two students who are attending my class, I could know their name. Because we are taking classes since 7 in the morning, and we would really appreciate who is attending our class. And please type in the chat box whosoever is attending the classes at the moment. Inhibition of enzyme activity is basically of three types: irreversible, reversible competitive, reversible non-competitive. Irreversible are the one which tightly go to the enzyme and does not let the ES complex to be made. But reversible competitive are the one which act resemble to the substrate and bind to the normal active site. With a reversible non-competitive, it does not bind to the active site, but inhibition is reversible in this case to other side. So irreversible inhibitors are the one, for example, arsenic snake, they tightly bind to the enzyme. They find where is the active site, so it doesn't let the ES complex form and no catalysis is happening. On the other side, reversible competitive inhibitors are the one with the enzyme inhibitors, also called as structure analogs. So they resemble like decoy of the substrate and occupy the enzyme active sites, and no reaction would occur in this case. But degree of inhibition depends on the relative concentration of enzymes and inhibitors in this case also. A non-competitor, they are the one uh, whose binding is weak and they attach to other site, not the active site, and hence it could be reversible. Then there are proteolytic enzymes which cleaves the peptide bonds in the protein. So like this, you have alanine, phenylalanine, glycine. So breaking of this peptide bond is possible. So this could be chymotrypsin is the one which breaks the peptide bonds of methionine, tyrosine, tryptophan, and phenylalanine. So some applications of enzymes in medicines, they could be used uh, to diagnose enzymatic level altered with the disease, heart attack, lactate dehydrogenase, creatinine phosphate, serum glutamate oxalate transferase (SGOT), pancreatitis, amylase, lipase, analytical reagents, enzyme used to measure another substrate, urea converted into ammonia via urease, and blood urea nitrogen measures, replacement therapy like administered genetically engineered B glucose sorbitolase for Gaucher disease. So that's it. I hope this was clear. This is a detailed version of enzyme. Good, Alex. Thank you very much. At least you are there listening me during this class. So I will continue for you, and I provide you the best content I could do till my last breath. Even though there are 20 students were shown in the group, it's a bit heartful that everyone was here for the certificates only, not for the studying. But nevertheless, whatsoever the condition is, we will continue with that. Next part, we will go into the core medicinal biochemistry, um, your core biomedicinal PPTs related to that, all the diseases. So just give me a break. So it's around 9 o'clock. We meet at 9:10, and then we start with uh, medicinal part. That is hypothyroidism. I'm coming within 10 minutes, so we start. You can take a break. Yeah, we'll also take a break. So hyperthyroidism. Okay. So I've shared actually the same stream. So I had shared the new link there. Maybe students could join there now. So to introduce hypothyroidism to the students, to understand the hypothyroidism and study various approaches to treatment. So basically we have a thyroid gland which regulates the metabolic functions of the body in virtually every cells, and everything from the brain to the skin is affected by the hormone made by the thyroid gland. And hypothyroidism, what happens is it slows you down, it makes you lethargic and fatigued. Your hair becomes brittle, your skin becomes dry, and you become cold much easier than the average person. So conditions where there is reduced production of thyroid hormone, categorized as a primary and secondary on the basis of its causes. Primary hypothyroidism occurs due to the improper functioning of the thyroid gland, may be classified as overt or subclinical hypothyroidism, and affect approximately 5% of individuals, with elderly women being most commonly affected. Then the secondary hypothyroidism, which occurs due to the inadequate stimulation of thyroid glands by the thyroid stimulating hormone, that is TSH, uh, and maybe due to the congenital or acquired defects in the pituitary or hypothalamus. They are rare and occurs in less than 1% of the individual. So in the primary hypothyroidism, the etiology, that is a thyroid dysfunction. So autoimmune thyroiditis, congenital absence or defects in thyroid tissue, thyroid removal by surgery, radio ablation by radioactive iodine, and destructions of thyroid tissues caused by infiltrative disorders. Then impaired synthesis of thyroid hormone, iodine deficiency, most common use, congenital enzymatic defects, drug mediated like thiamides, amidromine, lithium, amino, glutathamide, carbizomol could be dealt with it. And there is secondary hypothyroidism, that is a reduced secretion of your thyroid releasing hormones or thyroid stimulating hormone. There are hypothalamic disorders like tumor, infiltrative disorders. And hypopituitarism includes your mass lesions, pituitary surgery, pituitary irradiations, hemophagic epilepsy, and lymphocytic hypophysitis. And then clinical manifestations includes the symptoms: weight gain, dry skin, cold sensation, growth is retarded, poor concentration, constipation, dyspnea, syndrome, parasynesthesia, all these. And clinical manifestations includes the signs: cold peripheral extremities, puffiness of face, hands and feet, edema, hair loss, brittle nails, hypertension, slow relaxation of tendon reflex. So all of these signs of it. So what happens in the children's if this hypothyroidism is happening? There's a delayed growth in the children and delayed appearance of permanent teeth, delayed puberty, pseudohypertrophy of muscles. In order to find out the results, so there are two different tests we can do: thyroid stimulating hormone assay, which includes a primary test to establish the diagnosis, Um, Additional test includes estimation of T3 and T4, test for thyroid autoantibodies and thyroid ultra uh, sonography, serum cholesterol, increase in hypothyroidism. So overt hypothyroidism and subclinical hypothyroidism. T3, T4, TSH, T3, T4, TSH. Treatment, the goal is to mimic the normal physiological levels and elevate the signs, symptoms. It should be treated according to the individual person. So you can take LT4 replacement therapy or thyroid hormone therapy mixture of T3 and T4, or you can go with the replacement therapy of hormones also. So thyroid functions test in pregnancy. So it's very asked during the pregnancy time that how much is the level of your thyroid hormones are. So you have to maintain them during the whole uh, your thyroid hormone FT3, FT4, TSH, and the trimesters. There are three trimesters for each of them. So during the three trimesters, their value should be around this should be maintained. That is, in the non-pregnant female, it is 3.7 to 7.2. In the yeah, and on the right hand side in the pregnant, the range is shown. So they are pretty high in the pregnant state. And there's congenital hypothyroidism clinical features. In infants, it happen. Hypothermia, poor feeding, bradycardia, jaundice, and large posterior fontanel umbilical hernia. The children and adolescents with the growth failure, markedly delayed bone maturation, delayed eruption of permanent teeth, muscle pseudohypertrophy, delayed or precoccus puberty. So all these things are possible in the children's also. Comorbid conditions, so hyperthyroidism includes depression, infertility, autoimmune disorders, and hyperlipidemia also. So outcome in this, what we have studied so far, that students, you have understood the hypothyroidism, the cause, and various approaches to treatment. Thank you. Oh, I sent to the wrong group. This group, yeah. So that was about hypothyroidism. Now let's go to the next one. Jaundice. So here we can see a baby. The bilirubin hormone is getting less in that baby, and baby can't is a yellowish color, born by birth, and this is what jaundice is known as. So we will have you get to know about jaundice. What is jaundice is, various classifications and evolutions of jaundice, and study various approaches to its treatment. So jaundice is actually a French word which means jaune, that is yellow. So the French in the J initially is become J becomes Y. So it's a yellowish discoloration of tissue resulting from the deposition of bilirubin. So tissue deposition of bilirubin occurs only in the presence of serum hyperbilirubinemia. Is a sign of either liver disease or hemolytic disease. So carotenemia and the use of drug quinacrine are few other causes of yellow coloration of the tissue. And carotenemia can be distinguished from jaundice by sparing of salaria. In 4 to 37% patients treated with quinacrine, yellowish discoloration of skin is seen. So unlike carotenemia, quinacrine can cause discoloration of salaria. So under the steady state, normal metabolic conditions, about 3.9 milligram per kilogram or 250 350 milligram of bilirubin is produced each day from breakdown of heme from RBC in spleen and liver, and 1 gram of hemoglobin yields 36.2 milligram of bilirubin. So here we can see this is our cell which is producing red blood cells. This is a macrophage in the spleen, liver, or red bone marrow. So our RBCs death and phagocytosis is occurring, leading to the production of heme here, and further, globin and the amino acids are produced, and with them also your ferrous ion is being transferred, which is with the help of transferrin enzyme to the liver, which then being transferred the same iron from the liver towards your red bone marrow, and this Fe3 plus further change to with the help of globulin, vitamin B12, erythropoietin, form erythropoiesis in the red bone marrow, and all these bluish thing that you're seeing, this reddish thing is the seeing, it's happening in the blood. Then from there, erythropoiesis, it goes into the red blood cell, then circulation for about 120 days, yeah, and then it comes back here. And this cycle keeps going on from your liver, ferritin, to the red bone marrow, to the red RBC. Then this circulation will keep going for 120 days. So afterwards, after the 8th, it is also possible that the heme group will produce biliverdin, and this biliverdin will produce bilirubin, and bilirubin will goes into the liver, and with the help of bile, this will happen in the bile, it goes to the small intestine, and from small intestine it goes to bilirubin, and with the help of bacteria, urobilinogen is produced, and then it could go to the kidney to produce urobilin, to produce your urine, and then it could go into the stercobilin and goes into the feces, and then large intestine. So all these cycles that is working in our human body because of RBCs. Basically, RBC is the main reason for this. Everything is connected around the RBC and the heme group. And further, your liver, red bone marrow, kidney, small intestine, and large intestine, they are playing role. So in order to find out bilirubin concentration in your body, there is a test called as a Vandenberg test. So it's helpful in determining the type of bilirubin present in the serum. So normal serum gives a negative Vandenberg test, and it is performed using the diazo reagent. So when diazo reagent is added to serum containing conjugated bilirubin, a purple coloration is obtained within the 30 seconds. This is called direct positive Vandenberg reaction. Then etiological classifications of jaundice includes hemolytic, that is intracorpuscular defects, hereditary spherocytosis, hemoglobinopathies like sickle cell anemia, beta thalassemia, and is extracorpuscular defects like malaria, quinine, sulfonamides, burns, snake venoms, mismatched blood transfusion. Then hepatocellular, which produce viral hepatitis, malaria, typhoid, septicemia. And then toxics are the anesthetic agents like halothane, chloroform, uh, anti-tubular drugs, rifamycin, isoniazid, arsenic, mercury, gold could be the reason. And also cirrhosis, portal, and biliary. Third one is your obstructive jaundice. This is post-hepatic, like stones, stricture, parasites, congenital biliary atresia. So first is your congenital hyperbilirubinemia. That is your unconjugated. There are two types: unconjugated and conjugated. In the first one, disturbance of bilirubin transport, that is the Gilbert syndrome, and if there is disturbance of bilirubin conjugation, then it is a Crigel-Najjar syndrome. So disturbance in the excretion of bilirubin give rise to Dubin-Johnson, and the syndrome is known as Rotor syndrome. So this is the difference that we have just discussed, that is your hemolytic jaundice, haptic jaundice, and obstructive jaundice, pre-haptic, haptic, and post-haptic. So mechanism, first of all, in the pre-haptic, so we have excess breakdown of RBCs producing unconjugated bilirubin in the amounts more than healthy liver can conjugate and excrete. In the hepatic jaundice, you have inability of liver to efficiently conjugate and transport bilirubin into the bile due to the liver cell damage. In the obstructive jaundice, you have obstruction of the bile conducts so conjugated bilirubin can't flow through the biliary tract, freely disturbance in the increased serum conjugated bilirubin. And the types of serum bilirubin accumulated, in the first case is unconjugated, in the second both unconjugated and conjugated, in the third is conjugated. And if we do the Vandenberg test, so in the first case is indirect positive reaction, in second biphasic, the third one is a direct positive reaction. So urine bilirubin and urine urobilinogen. So in the first case is absent, in second is present, in third is present. And in this case it increases in the first your urine urobilinogen, and the second case it decreases, in third case it's also decreased. And fecal sterobilinogen markedly increased, reduced, absent. Fecal fat level is normal in the first case, increased and increased. Peripheral blood film is anemia, normal, normal. So these are the various medical perspective to see which kind of jaundice it is. Yeah. And some clinical characteristics. Is it brutus, tender liver, spleen, gallbladder, urine color, stool color? So we can see following results and able to answer them. This is also called breast milk jaundice. The few babies exclusively have breastfeed develop jaundice in second week of the life and may continue in the third month. So it is presumed to be due to inhibitory substances in the breast milk that interfere with the bilirubin conjugations like pre-cannabidiol and free fatty acids, and bilirubin level of over 20 milligram deciliter may be attained, and temporary interruption of breast milk feed reduces the serum levels of bilirubin. Then pathological jaundice in newborns, so they appears within 24 hours of age. They increase the bilirubin more than 5 milligram per deciliter per 24 hours. Total serum bilirubin is 15 milligram per deciliter, and direct bilirubin would be more than 2 milligram per deciliter. Hemolytic jaundice in newborn. This condition results from the incompatibility between maternal and fetal blood groups. So rhesus positive fetus may produce antibodies in rhesus negative mother, and rhesus incompatibility first children often escapes, so subsequent children would be affected. Sometimes the child is born with severe hemolytic disease, often referred to as erythroblastosis fetulis. So when the blood level is more than 20 milligram per deciliter, the capacity of albumin to find bilirubin is exceeded. So we have learned about jaundice, some understand the classification of jaundice, and we get to know about the various approaches to the evolution and treatment. So that was about jaundice, its various in detail biochemistry with diagnosis, etiology, 3 different types, where it is working, how it is working, what other treatments we can do. So we have discussed in quite detailed manner about jaundice also. So give me a 5 minutes break. At 9:30 we start the last topic, that is your LFT. So then we have mainly the gel electrophorces. I will show you the Western blots in them. So we go with that, and the rest of your part will be over. thyroid function, LFT, jaundice, protein hormones, nucleic acids, lipids, amino acids. So we have covered your whole topic in the 6 hours. So just your that will be left. So yeah, we will start soon then. Give me a moment. 5 minutes. Taking break. So liver function test. So let's continue with the liver function test. So liver is the largest organ of the body, as we know, around 1.5 kg, and it's the kitchen of our body, and it's take care of our carbohydrate metabolism. So in fat state, when we eat it, glycogen synthesis takes place, and excess glucose is converted into fatty acids and tags, which get incorporated into VLDL, transported to adipose tissue. And in fasting state, glucose concentration is maintained by glycogenolysis and gluconeogenesis. So in protein metabolism, synthesis of albumin, various plasma proteins except immunoglobulins, and most of the coagulation factors like fibrinogen, prothrombin, like 5, 7, 9, 10, 11 should be taken care. And then LFT, total bilirubin from 0.2 to 0.8 milligram per deciliter. Conjugated bilirubin is 0 to 0.2, total 6 to 8, albumin 3.5 to 5. These are the liver function test in the normal, and coagulation factor should be around 11 to 12 seconds. So enzymes that are produced in this case are SGPT, SGOT, ALP, GGT, and nucleotides. So remember, whenever you want to go for liver tests, so you will mainly they are giving these values: SGPT, marker enzyme for liver disease; AST is SGOT, alkaline phosphatase, gamma glutamyl transferase. Yeah. So special tests are bile acid level, blood ammonia, alpha 1 antitrypsin, alpha 1 fetoprotein, hepatitis markers, immunoglobulins, ceruloplasmin, ferritin. Total serum bilirubin includes 0.2 to 0.8 milligram per deciliter, and conjugated includes bilirubin 0.2 less than 0.2 milligram per deciliter, and unconjugated bilirubin 0.2 to 0.6 milligram per deciliter. And we do mainly Vandenberg reactions. This is to check normal serum gives a negative Vandenberg reaction, and the positive serum will give a positive reaction. So in this, mainly equal volume of sulfonic acid and dilute hydrochloric acid and sodium nitrate is added. Then that sulfonylic acid reacts with the bilirubin to form purple colored, that is azobilirubin. So conjugated bilirubin gives purple color immediately on addition of the reagent. Indirect positive would be purple color develops only reagents and methanols are added. So biphasic in this, your purple color develops an addition of reagents and addition of methanol interferes the color and elevation of both unconjugated and conjugated bilirubin. So indirect positive is the hemolytic jaundice, direct positive is the obstructive jaundice, and biphasic is the hepatic jaundice. So three different things could be coming out from it. And bilirubin in urine, normally bilirubin is absent in urine, and conjugated bilirubin being water soluble is excreted in the urine in obstructive jaundice, and this can be detected by the Foctet test. And urine urobilinogen normally trace amounts is present, and in obstructive jaundice no urobilinogen is present in urine. Because bilirubin cannot enter intestine, so presence of bilirubin in urine and absence of urobilinogen in urine is seen in obstructive jaundice. So in hemolytic jaundice, increased production of bilirubin causes increased formation of urobilinogen, which appears in urine. So note, increased urobilinogen in urine and absence of urobilinogen in urine is seen hemolytic jaundice here. And the fecal urobilinogen gives normal about 300 milligram, an increase in hemolytic jaundice in which color of feces is dark. And in obstructive jaundice, urobilinogen is not excreted through feces, so color is feces is pale. So I remembered I had this jaundice in my childhood. So my fecal color used to be white color. The yellowish color, this bilirubin is lost in jaundice. It's a horrible, horrible time. The serum albumin, about 10 to 12 grams of albumin is synthesized in liver daily. Its estimation is very valuable in assessing the chronic liver disease, and low serum albumin level is commonly observed in severe liver diseases. Prothrombin time, normal 11 to 12 seconds. It is prolonged in severe pericardial liver disease due to decreased synthesis of prothrombin. Vitamin K is required for the synthesis of prothrombin. Vitamin K deficiency can also lead to the prolonged PT. So plasma proteins of diagnostic values in liver disease, that is your albumin, chronic liver disease, change in concentration, gamma globulins, cirrhosis, especially autoimmune, increased change in concentration, alpha 1 antitrypsin, cirrhosis due to alpha 1 antitrypsin deficiency is decreased. And yeah. Then transminase with the SGPT and SGOT 3 to 15. The SGPT value should be and SGOT should be 4 to 17. And ALT is primarily localized to the liver, so it is a marker enzyme of the liver. It's present in the cytosol of the hepatocytes and it's quite wide variety of tissues like heart, liver, skeletal muscles, kidney, brain. And AST is present in both in cytosol and mitochondria of your hepatocytes. So liver contains both enzymes but more of ALT. So estimation is very useful in assessing severity and prognosis of your liver, how much it is bad, and very useful in screening the outbreak of infective hepatitis. So if it is highly elevated 20 times more, viral hepatitis is there. So moderately elevated is 3 to 20 times. And then it's a chronic hepatitis. And there's also alcoholic hepatitis, autoimmune hepatitis, acute biliary tract obstruction. So alkaline phosphatases, ALP, which is 3 to 13, a family of zinc metalloenzymes with a serine at the active center. They release inorganic phosphate from various organic phosphatases. In the liver, it is found in microvilli of bile caniculi and the sinusoidal surface of the hepatocytes. Other important source of ALP is bone, and ALP is highly elevated in obstructive jaundice and bone disease like rickets. Then gamma glutamine transcriptase, normal level is around 10 to 15 units per liter. So it is a membrane-bound glycoprotein which catalyzes the transfer of gamma glutamyl group to the other peptides and very useful in diagnosis of obstructive jaundice. It is a microsomal enzyme. Serum GGT is highly elevated and enzyme is inducible enzyme. Then nucleotides, which is normal 2 to 15, it is elevated in obstructive jaundice, and is advantages is that it's not elevated in the bone diseases. To test the detoxification function of liver, hyperuric acid test could be done. In this, liver is put in the benzoic acids combined with the glycine, and procedure is 6 gram of sodium benzoate is given to the patient. Urine is collected up to 4 hours, and hyperuric acid excreted in urine, and 6 gram of sodium benzoate forms, making 7.5 gram of hyperuric acid. And 60% of sodium benzoate is excreted in normals, and decreased hyperuric acid excretion 3 gram indicates hepatic damage. So bilirubin is a normal or mild increase, total proteins normal, albumin may decrease, globins increase, ALP highly elevated, and BS purification is 45 minutes that is increased. Alcoholic hepatitis, the bilirubin is mild, ALP will be elevated, ALT will be mild, and GGT will be highly elevated. So each conditions, acute hepatitis, chronic, cirrhosis, cholestasis, malignancy. So all these values are shown, your albumin, alkaline phosphatase, aminotransferases, bilirubin, gamma globulin, prothrombin time. Each values has been explained. It should be normal or high, how it should be. So that was core details, quite chemically corrective LFT part. So by this, actually, we are done with the today's lecture. But at the end, I want to teach you something about Western blotting. So that was left from yesterday. gel electrophoresis basically. So southern, northern, we can do it later. Mainly the western. So in the western blotting, it's an analytical technique used to detect specific proteins in a given sample. Uh, so it's mainly done to check the how much protein is present in it, and it's done with the help of electrophorized on an SDS page, and electrotransferred on nitrocellulose membrane, and then we had primary and secondary antibody in order to find out the answers. So yeah. So basically, in this, as we have discussed, so I will. This is the nitrocellulose membrane having the basic protein. Then we had the primary antibody. Then we had the secondary antibody, and then on the top we had our SOFOS, XFOS, to detect the location of a secondary antibody for our eyes. So let's watch one video about that. Then we'll wrap up this session.
In this video, you will learn how to perform a Western blot. A Western blot can be used to identify specific proteins in a sample and provide information about the protein size and relative abundance in the sample. First, fill a tray with blotting buffer. You will be using this buffer to equilibrate your gel prior to starting the Western blot. Next, remove the gel from the gel cassette using the opening key. Line up the arrows on the opening lever with the 4 arrows on the cassette to open the cassette. After trimming the top and bottom of the gel with a straight edge, equilibrate the gel in the tray with blotting buffer for 15 minutes on a rocking platform. Pre-soak fiber pads in blotting buffer so that they are thoroughly soaked. To make a blotting sandwich, obtain a container large enough to fit the gel holder and add enough blotting buffer until the container is filled approximately 1 centimeter deep. Place the gel holder cassette in the container with the black side down and immersed in the buffer, and the white side up and out of the buffer. Lay 1 fiber pad flat on the black plastic. Next, wet a piece of blotting paper and place it on top of the pad. Be careful to avoid any bubbles between the pad and the paper, and make certain the buffer covers the paper. Take the gel and carefully place it squarely onto the blotting paper. Again, being careful to avoid any bubbles between the gel and the blotting paper. Next, you will be applying a piece of nitrocellulose membrane. Remove the protective sheet from the membrane and wet the membrane with blotting buffer. Carefully place the membrane squarely on the gel. Avoid moving the membrane once placed on the gel, as proteins will begin to blot immediately. Using a roller, remove any air bubbles between the gel and the membrane. Place a second sheet of wet blotting paper on top of the nitrocellulose membrane. Place a second wet fiber pad on top of the blotting paper. Fold the clear plastic side of the gel holder over the sandwich and clamp it to the black plastic side by sliding over the white clip. This tight fit will squeeze the sandwich together. Insert the gel holder into the inner module. Make certain that the black side of the gel holder is next to the black side of the module. Place the inner module into the electrophoresis chamber. Add a frozen cooling unit and fill the chamber with blotting buffer to the level of the white clip on the gel holder. Place the lid on the electrophoresis chamber. Connect the electrical leads to the power supply, making sure the connections are correct, red to red and black to black. Turn on the power supply and run the blot at 20 volts. If a timer is available, set it for 2.5 hours. When the run is complete, turn off the power supply and disassemble the electrophoresis chamber and remove the inner module. Open the module and place it in a container filled with blotting buffer with the black side down. Starting with the first fiber pad, remove each layer until you reach the nitrocellulose membrane. As you remove the membrane, note that the proteins have been transferred from the gel to the membrane. Note that the kaleidoscope pre-stain standards have been transferred and can be seen on both sides of the membrane. You can also see that there are no longer any proteins on the gel. Immerse the membrane in 25 milliliters of blocking solution and incubate it for 15 minutes at room temperature on a rocking platform. Pour off the blocking solution. Add 10 milliliters of primary antibody. Incubate for 10 to 20 minutes on a rocking platform. The platform should be set at a faster setting to ensure constant coverage of the membrane. Pour off the primary antibody. Rinse the membrane quickly in 50 milliliters of wash buffer and then discard the wash buffer. Add another 50 milliliters of wash buffer to the membrane and let it wash for 3 minutes on the rocking platform at a medium speed setting. Discard the wash buffer. Add 10 milliliters of secondary antibody and incubate the membrane for 5 to 15 minutes on the rocking platform at a fast speed. Pour off the secondary antibody. Rinse the membrane quickly in 50 milliliters of wash buffer and discard the wash buffer. Add an additional 50 milliliters of wash buffer and wash the membrane for 3 minutes on the rocking platform on a medium speed setting. Discard the wash buffer. Add 10 milliliters of substrate. Incubate the membrane with the substrate for 10 to 30 minutes with either manual shaking or on a rocking platform. Watch for the color development. Once the colors have developed, rinse the membrane twice with distilled water and blot dry with a paper towel. Air dry for 3 to 60 minutes and then cover in plastic wrap for storage.
So that was the gel electrophoresis. So we are done for today. Thank you very much everyone for being there. Streaming went fine. That's good. And yeah, just give your comments on the yesterday I have shared with you, I guess, on the Edufabrica thing. Just give your honest comment. How did you like the? Okay then. Take care. Bye bye. And if you, yeah, stay safe. Take care.